Okay, let's unpack this. When we talk about injury, in the medical sense, most people, they instantly picture something, visible,
Yeah.
traumatic.
A broken bone, a cut.
Exactly, or
Right.
collision. But today, we're dealing with the kind of damage that's, well, quieter,
Yeah.
more molecular, but it's actually far more disruptive to clinical medicine, to pharmacology,
Mm-hmm.
chemical and toxic injury.
Mm.
And this isn't just about those rare, dramatic poisoning stories you see in the news. This is fundamental, a really pervasive problem.
It truly is. I mean, chemical injury is still a surprisingly frequent clinical challenge. Could be environmental exposure, could be a reaction to a drug someone needs.
Right.
And if you look at, say, pharmaceutical development, the whole landscape of safety and economics, toxicity, especially unexpected toxicity, is a huge limitation globally.
So a drug looks promising, goes through trials.
Yeah, and then late in clinical trials, it fails. And the reason, so often it traces right back to some kind of unpredictable cellular toxicity.
That immediately brings one organ into focus, doesn't it? The body's main processing center, which unfortunately kind of becomes the body's chemical punching bag.
Yeah.
I'm talking about the liver.
Precisely. The liver is the prime target. And toxic liver injury is, tragically, often the exact reason a therapeutic drug gets pulled from the market, or maybe even terminated during development.
Why the liver, specifically?
Well, think about it. It's the body's primary metabolic processing center. Its job is biotransformation, breaking down foreign substances, xenobiotics we call them.
To neutralize them.
Yeah, neutralize them, make them easier to excrete. But that very function, that essential cleanup role, is exactly what makes it so vulnerable.
Okay, so if the liver's the factory, and our mission today is to figure out how that factory gets sabotaged, we need to get beyond just saying chemicals are bad. We need to understand how the attack actually happens at the cellular level. So the goal today is really to distill this huge field of toxicology down to its core mechanisms. What are the, say, two major distinct ways chemicals attack our cells?
Yeah, we can really simplify the mechanisms. Think of it as two fundamentally different pathways for chemically induced injury.
Okay.
Pathway one is the chemical acting directly. It immediately attacks, combines with crucial cellular components just as it is in its original form. Think of it like a direct chemical assault.
The direct hit. And pathway two.
Pathway two is, well, it's much more nuanced, almost like biological friendly fire, you could say.
Friendly fire. How so?
Because the initial chemical is often relatively inactive, maybe even harmless, sort of like a pro-drug. But when our body, primarily the liver, tries to clean it up, tries to process it to make it excretable,
Yeah.
that cleanup crew accidentally creates a highly reactive, unstable fragment. That's the toxic metabolite.
Wow. Okay. So the body makes the poison itself, trying to get rid of the original substance.
Exactly. And these two pathways, they lead to entirely different patterns of cellular destruction. And critically, they need completely different treatment strategies.
That's a really powerful distinction. Okay, let's dive right into pathway one then. Direct cellular toxicity. You said this means the chemical itself, right when it enters the body, is the immediate attacker. No metabolic conversion needed.
Correct.
So what does this direct chemical assault actually look like? Down at the microscopic level.
So indirect toxicity, the chemical molecule itself, its structure is just predisposed to immediately bind with or react with critical macromolecules inside the cell. Proteins, lipids, things like that.
It causes instant disruption.
Instant disruption. The damage happens right on contact or as soon as it's absorbed. And it often just overwhelms the cells that first encounter the chemical in high concentration.
Okay, let's make this concrete. Let's start with a classic example you mentioned, attacking the cell structure itself. Mechanism A, membrane attack and transport shutdown, using mercuric chloride poisoning. That stuff is notorious for its quick, devastating effects.
Right. Mercuric chloride is really a textbook illustration. It shows exactly how a chemical can just shut down essential cellular architecture. Mercury and other heavy metals like it has this high affinity for. It binds directly to the sulfhydryl groups.
Sulfhydryl groups. What are those?
They're chemical functional units. They contain sulfur and hydrogen, and they're absolutely essential for the proper folding and function of proteins, especially those proteins embedded right in the cell membrane.
Okay, so mercury basically gums up those specific protein structures. If it does that, what are the primary biological systems that just immediately fail? What does that cascade look like for the poor cell?
Well, the consequence is twofold. And it's catastrophic.
Yeah.
First, that binding makes the membrane lose its structural integrity. It gets leaky, increased permeability.
Okay.
Second, and this is really critical, it inhibits essential functions like ion transport. Think about the crucial Na plus K plus ATPase pump, the sodium pump.
Yeah, that maintains the cell's internal environment.
Exactly. It's responsible for that. And it relies heavily on proteins that have functional sulfhydryl groups.
So when the mercury binds, that pump just shuts down instantly.
Instantly shuts down. And that means the cell can no longer actively push sodium ions out.
Help me understand the consequence there. If the cell can't regulate sodium?
Well, sodium starts building up inside the cell.
Mm-hmm.
And water follows sodium, right? Osmotic pressure.
Okay.
So this leads to rapid, uncontrolled cellular swelling. The cell's entire volume regulation system just collapses.
It literally bursts.
The swelling stresses the membrane to the breaking point, and that leads very quickly to cell lysis, the cell bursting open in irreversible necrosis, cell death.
And this specific mechanism, this binding to sulfhydryl groups, it determines which organs get hit the hardest, right? It's very localized destruction.
Exactly. The damage concentrates in the cells that have the highest exposure, or the ones actively involved in absorbing it, filtering it, or excreting the chemical.
Makes sense.
So with mercuric chloride, you see this acute, catastrophic damage in the cells lining the gastrointestinal tract. That's where it's absorbed first.
Right.
And critically, the kidney tubules. They're the filtration and excretion machinery, and they end up concentrating the toxic substance before it can really be processed elsewhere. So the damage is swift and it's focused.
Okay, that's a structural attack.
Yeah.
Now let's look at a different type of direct attack you mentioned. Immediate energy shutdown. Mechanism B. And the classic, honestly, terrifying example here is cyanide poisoning.
Okay.
Instead of wrecking the structure, this poison just turns off the lights.
Yeah, cyanide is maybe the most efficient chemical killer precisely because it targets the cell's universal power supply. It goes straight for the engine room, the mitochondria.
Okay.
Cyanide directly and non-competitively poisons mitochondrial cytochrome oxidase.
Why is that specific enzyme, cytochrome oxidase, so critical? Can you map that out for someone maybe learning this?
Sure. Cytochrome oxidase, sometimes it's called complex 4. It's the final, absolutely essential step in the electron transport chain, the ETC.
Okay, the ETC.
Think of the ETC like a cellular production line. Electrons get passed down a series of protein complexes, and energy is harvested slowly along the way.
Right.
The ultimate goal is to use oxygen to accept those electrons at the very end. And that process drives the production of massive amounts of cellular energy, ATP, through oxidative phosphorylation.
So if cytochrome oxidase, the final step, is poisoned by cyanide, what happens to energy production?
Cyanide binds incredibly tightly and instantly to the ferric iron atom right within that cytochrome oxidase enzyme.
Wow.
And that stops the flow of electrons immediately. The entire ETC production line just jams up solid.
So no final step means?
No final step means oxygen cannot be utilized by the cell to generate ATP. The cell's main power factory goes dark instantly.
That is just catastrophic energy failure. I mean, without ATP, the cell can't run its sodium pumps, like we just discussed.
Can't run pumps, can't synthesize necessary proteins, can't maintain any kind of homeostasis. It's over.
Which cells get hit first and hardest in that scenario?
The ones with the highest energy demands, the highest metabolic rate. So that means the neurons in the central nervous system.
Brain cells.
Brain cells, yeah. And cardiomyocytes in the heart, muscle cells of the heart.
So the effects are incredibly fast.
Within minutes. The systemic inability to use oxygen, even though there might be plenty of oxygen delivered in the blood, that leads to rapid neurological failure, seizures, coma, and then cardiorespiratory arrest. The speed really highlights the instant direct nature of this kind of toxicity.
It's important, I think, for us to pause here and realize something. We're talking about poisons like mercury and cyanide, really scary stuff. But this whole concept of direct cytotoxicity, it's not just the domain of toxins, is it? Medicine actually uses this mechanism too.
Absolutely. That's a great point. Many common drug classes are actually designed to operate through controlled, direct cytotoxic effects.
Like what?
Well, think about certain antibiotics. They're designed to disrupt bacterial cell wall formation. That's basically a targeted membrane attack, right?
Yeah.
Or, more prominently, anti-neoplastic chemotherapeutic agents, chemo drugs.
For cancer.
Exactly. They often work by introducing a chemical that we know will bind directly to, say, DNA, or maybe key enzymes that are essential for rapid cell division. The goal is to induce targeted cell death in tumors.
So it shows that direct injury is this powerful, fundamental biological mechanism used by poisons, but also harnessed by medicine.
Precisely. It's a tool, a mechanism, can be destructive, can be therapeutic.
Okay. So pathway 1, fast, focused. The parent chemical is the villain. Got it. But now let's move to the path that feels, I don't know, conceptually more challenging, pathway 2, where the injury comes from our own body's essential, but sometimes tragic attempt to clean up the mess.
Yes. And what's fascinating here, as I mentioned, is this element of betrayal almost.
Betrayal.
Because of the parent substance, the chemical that first enters the body, it starts out relatively benign, or at least manageable. As we established, most toxic chemicals, or xenobiotics, they're lipophilic, fat-soluble. Now, for the body to get rid of them, usually through urine, they have to be made water-soluble.
Yeah.
This process is called biotransformation. It's absolutely essential for survival. But, and here's the catch, in the case of many toxins, the intermediate products created during this process are hyperreactive.
So the cleanup crew accidentally creates the toxin.
That's exactly it. The body's own cleanup crew makes the problem worse.
That sounds like the biological equivalent of trying to clean a really dusty antique using, I don't know, acid. The intention's good, but the intermediate step is highly corrosive.
That's a pretty good analogy, actually.
So where does this crucial and sometimes catastrophic conversion actually happen in the body?
It happens primarily in the liver. Specifically, it's localized in the smooth endoplasmic reticulum, or ER.
The smooth ER.
Yeah, which is packed, absolutely rich in these enzymes we call the cytochrome P450 mixed function oxidases.
The P450 system. You hear that mentioned a lot in drug metabolism. It's often described as the body's main detox factory.
It is.
But it sounds like it's often the factory that manufactures the trouble. Can we detail this faulty grinder analogy you used earlier?
Yeah, the P450 system is incredibly sophisticated and necessary, don't get me wrong. Its function is to kick off detoxification, usually through a two-phase process.
2 phases, okay.
Phase I reactions, which are mediated by these P450 enzymes, are usually things like oxidation, reduction, or hydrolysis. These reactions basically stick a polar functional group, like a hydroxyl group, onto the lipophilic chemical, makes it slightly more water-soluble.
So phase I is the modification phase, getting it ready, and this is where the trouble starts.
Absolutely. This is often the danger zone. The chemical intermediates created in phase I, they're often unstable, highly reactive. They might possess a transient, unpaired electron, which is the very definition of a free radical.
Okay, so they're unstable. What's supposed to happen next?
In a perfect world, these reactive intermediates immediately move on to phase II. That's the conjugation phase.
Conjugation, meaning?
Meaning they get linked up, conjugated to a large water-soluble molecule, like glucuronic acid or sulfate. This usually neutralizes them completely and makes them really easy for the kidneys or bile to excrete.
Right. So the problem arises when the rate of that first step, the phase I conversion, exceeds the speed or the capacity of the second step, the phase II neutralization.
That is the absolute core of indirect toxicity. If the P450 system, acting like that sophisticated but maybe overwhelmed grinder, produces these highly reactive phase I fragments the toxic metabolites faster than phase II can handle them.
They escape.
They can escape the ER before phase II neutralization happens, and then they're loose in the cell, free to initiate destructive processes.
Okay, so once these unstable fragments, these free radicals, are created and escape, how do they inflict the actual damage? What's the main mechanism of injury in pathway 2?
Well, the most common and damaging mechanism is through the formation of more free radicals, often specifically reactive oxygen species, or ROS. That initial P450 oxidation process itself can spin off these highly volatile, unstable molecules.
Okay, and once created, what do these ROS do?
They initiate a devastating process called lipid peroxidation.
Lipid peroxidation. Can you walk us through that? It sounds like, I don't know, chemical destruction that spreads, like a chain reaction.
It is precisely a chain reaction. Free radicals are desperate for stability. They want to steal an electron from somewhere.
Right.
And the nearest large, accessible, and vulnerable targets, they're the fatty acids that make up our cell membranes, the mitochondrial membranes, the ER membranes themselves.
So the free radical attacks the fat in the membrane.
It rips an electron off a fatty acid chain in the membrane. It stabilizes itself, sure, but in doing so, it turns that previously stable fatty acid into a new free radical.
Oh, wow. So one destructive radical literally creates another one, and the damage just spreads exponentially across the membrane structure.
Exactly. It's an autocatalytic chain reaction. This destructive oxidation degrades the lipid bilayer, severely compromising the structural integrity and the function of all these vital cellular compartments.
So if the membranes get damaged,
Compartmentalization fails. Organelles can rupture. The whole system collapses from within. And additionally, these toxic metabolites can also cause direct covalent binding.
Covalent binding?
Yeah, they can physically, permanently bond themselves to crucial cellular proteins and lipids, basically inactivating them, adding insult to injury, further compromising cell function.
Let's look at a real-world example. The historical, kind of archetypal case study for this mechanism. Carbon tetrachloride, CCl4.
Right, CCl4. It was once universally used as a cleaning solvent, especially in dry cleaning, also fire extinguishers.
Right.
It is the absolute textbook example of P450-mediated injury.
And CCl4 itself isn't the main problem.
CCl4 itself is relatively stable, relatively inert, but when it gets into the liver, the cytochrome P450 system, in its attempt to detoxify it, to make it soluble, it converts CCl4 into the highly reactive free radical CCl3. That's the trichloromethyl radical. That little dot signifies the unpaired electron, extremely reactive.
So the creation of that radical, the CCl3, that's the moment of catastrophe.
That is the trigger. This radical immediately drives massive lipid peroxidation. It causes extensive, severe damage, particularly to the ER membrane first, where it's generated, and then it spreads to the mitochondrial membrane.
And the result for the liver cell?
Acute liver cell death, massive central lobular necrosis, typically. The cell's internal machinery is basically chemically liquidated by this runaway chain reaction. Understanding CCl4 was so crucial because it really proved that the body's own detoxification system could be the actual chemical trigger for the poisoning.
It's really unsettling, isn't it? To think that chemical once used to clean our clothes was basically a biological time bomb, just waiting for our own liver to pull the trigger.
It is.
But the danger isn't just limited to old industrial chemicals. The exact same mechanism plays out with a very common, widely available pain reliever, acetaminophen, known as paracetamol in many parts of the world.
Yes. Acetaminophen toxicity is, you could argue, the most clinically relevant example of this indirect injury pathway today. It's actually the leading cause of acute liver failure in many developed countries.
Wow. Leading cause.
Yes, and it perfectly illustrates this threshold concept of P450 liability.
Okay, how does acetaminophen fit into that phase I, phase II paradigm we talked about?
So the vast majority, maybe 90% of a normal therapeutic dose of acetaminophen is safely processed. It goes straight through phase II conjugation, glucuronidation, and sulfation, makes it harmless, easily excretable, no problem.
Okay, 90% is safe.
But a small portion, maybe 5%, maybe 10%, gets shunted through the P450 system, specifically an enzyme called CYP2E1, and that enzyme is highly inducible.
Inducible meaning?
Meaning its activity can be increased by certain things, like chronic alcohol use, for instance.
Okay, so when that small portion goes through P450, through CYP2E1, that's when it creates the toxic metabolite.
Exactly. It creates something called N-acetyl-P-benzoquinone imine, usually just called NAPQI.
NAPQI.
NAPQI is an incredibly reactive electrophile. That is the toxic metabolite. It's primed and ready to bind covalently to liver cell protein and cause damage.
But normally that small amount doesn't cause a problem.
Yeah. Normally, no. Because fortunately, the cell has a built-in natural antioxidant shield. It's called glutathione.
Glutathione. Heard of that?
Yeah. Glutathione rapidly neutralizes NAPQI by conjugating with it. It sacrifices itself, basically.
So under normal dosing, that glutathione shield easily handles the small amount of NAPQI that gets generated. The metabolite is neutralized, excreted, end of story.
Precisely. The system is designed to handle that low level. But when the dosage of acetaminophen is super therapeutic, meaning an overdose,
Way too much is taken.
way too much, then the rate of NAPQI production by that P450 enzyme just completely overwhelms the rate of glutathione conjugation. The cell's glutathione stores get completely depleted, used up.
And once that shield is down,
the unneutralized NAPQI is free to run rampant. It causes extensive covalent binding to essential mitochondrial proteins, other cellular proteins, leading to rapid, irreversible liver necrosis, cell death.
that's why immediate treatment is so critical in an overdose.
Absolutely. That's why giving something like N-acetylcysteine, or NSC, is the standard treatment. NAC is a precursor for glutathione. You're essentially trying to rapidly rebuild that antioxidant shield as fast as possible to soak up the NAPQI before it causes lethal damage.
That comparison, CCl4 and acetaminophen, it really brings into sharp focus the delicate balance our bodies maintain. It's not just about the amount of chemical, but also the state of our detox system.
Exactly. Capacity matters hugely.
Okay, so having detailed the mechanism of the damage direct binding versus this P450 activation, Let's maybe transition now to the application.
Mm.
What are the future challenges this understanding poses for modern medicine and toxicology research? Hashtag #TechForMar. The clinical and research implications. Connecting the dots.
Right. If we connect this detailed mechanistic understanding back to the bigger picture,
Yeah.
well, the pathways we've just outlined, they really inform every frontier of drug development, patient safety, toxicology research.
Mhm.
We're moving from just observing injury to actually trying to predict it.
Okay, let's start with one of the most pressing clinical issues, that huge variance in drug safety. Why does the exact same dose of a drug injure one person severely, maybe even fatally, but not affect another person at all?
Yeah, that takes us straight to challenge one, predicting idiosyncrasy and susceptibility.
Yeah.
And it's deeply, deeply rooted in genetic differences that affect that very P450 system we just discussed.
Ah, genetics again.
The efficiency of those P450 enzymes, it's determined by genetic polymorphism. We all have slight variations, different versions of the genes that code for these enzymes.
So this is pharmacogenomics, right? Where your personal DNA dictates how well or maybe how poorly you process a particular substance.
Yeah.
What are the major ways this P450 variability shows up?
We can broadly categorize people based on their metabolic rate for a specific P450 enzyme, let's say CYP2D6, which is a really important one. You basically have three main groups.
Okay.
First, you've got poor metabolizers, or PMs. Their P450 enzymes for that pathway might be inactive or just nearly absent.
So what happens if they take a drug cleared by that enzyme?
Well, the drug hangs around in their system way too long. It doesn't get broken down efficiently. This can lead to extremely high plasma concentrations and potentially severe dose-dependent toxicity, even if they're taking what's considered a standard dose.
Because they just can't process the drug fast enough to get rid of it.
Exactly. Then second, you have extensive metabolizers, EMs. That's considered the normal group. Most standard drug doses are kind of calibrated for these individuals.
Okay. And the third group?
The third group is ultra-rapid metabolizers, or UMs. These individuals have multiple copies of the enzyme gene, so their processing machinery is incredibly fast, super efficient.
And the ultra-rapid metabolizers? They face a problem that relates directly back to pathway 2, don't they? The metabolite conversion pathway.
Precisely. If the drug happens to be a prodrug, remember, a chemical that requires P450 activation to become therapeutically active, then UMs can convert it so fast that they generate an excessive amount of the active compound. This could lead to rapid, maybe dangerously strong therapeutic effects. Or, even worse, if the metabolite itself is toxic, like NAPQI from acetaminophen.
They'd make too much toxic stuff too quickly.
Exactly. They could overwhelm their detoxification shield, like glutathione, much faster than a normal person would, increasing their risk of toxicity.
Can you give us an example where this variability is really critical in practice?
Yeah, a classic one is the opioid painkiller codeine.
Codeine?
Codeine is actually a prodrug. It has virtually no pain-relieving effect on its own. It must be converted by that CYP2D6 enzyme into morphine to work.
Ah, I didn't know that.
So think about it. An ultra-rapid metabolizer. They take a standard dose of codeine, and their body rapidly converts it into massive, potentially lethal doses of morphine. Very dangerous.
Wow. And the poor metabolizer.
The poor metabolizer gets almost no pain relief at all because they simply cannot convert the codeine into morphine effectively.
So the same pill has wildly different effects based purely on genetics.
Wildly different. One person gets no effect, another gets a potentially fatal overdose effect from the same standard dose. This individual variance dictates who's vulnerable to drug toxicity and who experiences drug failure. And this understanding, this is what's driving the whole field of personalized medicine.
Using genetic tests before prescribing.
Exactly. Using genetic tests to predict susceptibility, predict response before we even give the first dose, tailoring therapy to the individual's P450 profile.
That's a huge implication for clinical practice, for safety. Now let's pivot a bit. Let's talk about cellular defense strategies. We saw that regardless of the mechanism, whether it's direct injury like cyanide hitting the mitochondria or indirect injury like CCL4 causing free radical damage that hits the mitochondria, those mitochondria seem to be prime target either way.
Absolutely.
So protecting them feels like, I don't know, the ultimate barrier against irreversible cell death?
It really is. That's challenge two. Protecting the mitochondria. Mitochondrial collapse, mitochondrial failure. That's often the point of no return for cell necrosis.
So in the context of that indirect injury pathway, pathway 2, where free radicals are the main agents of destruction, the therapeutic focus has to be on neutralizing those radicals before they start that lipid peroxidation chain reaction.
That's the goal. You have to stop it before it starts, or at least very early on.
So how are researchers actually trying to get antioxidants or protective agents into the exact place they're needed, like deep inside the cell, maybe even specifically targeting the mitochondrial inner membrane to neutralize those ROS?
Yeah, it's incredibly complex because remember, the cell membrane and the mitochondrial membranes themselves, they're designed to be highly selective barriers.
Right, they don't just let anything in.
Exactly. So
Yeah.
just, injecting a generalized antioxidant into the bloodstream often results in poor cellular penetration, or maybe the antioxidant gets degraded rapidly before it even reaches the site of damage inside the cell.
So you need better delivery.
Right. Future advancements are focusing heavily on targeted delivery systems.
Are we talking about things like nanotechnology here? Liposomes, nanoparticles?
Precisely. Researchers are developing really clever molecular systems. Could be based on specific peptide sequences that mitochondria recognize, or nanoparticle constructs designed to be actively taken up by the cell and then specifically home in on the mitochondria.
Like a tiny guided missile carrying antioxidants.
Kind of, yeah. The goal is to deliver highly concentrated antioxidant agents. Maybe precursors like the N-acetylcysteine we talked about for acetaminophen overdose, or maybe entirely novel compounds designed to scavenge specific radicals like superoxide or hydroxyl radicals, deliver them right to the source of the problem. Block that destructive chain reaction of lipid peroxidation right where it starts.
It sounds like we're trying to develop a targeted molecular shield that we can deploy right inside the cell's engine room, the mitochondria.
That's a good way to put it.
This brings us back, though, to that big obstacle you mentioned earlier, the one that haunts drug development, the liver dilemma.
Yeah, the clinical urgency for all this research, understanding P450, finding ways to protect cells, it's immense. Because toxic liver injury, often caused by these unexpected P450 metabolic accidents, is what consistently derails promising drug development projects.
After huge investments.
After years of work, billions of dollars spent. And the failure often only becomes apparent really late, like in phase 2 or phase 3 human trials.
So how is research trying to solve that prediction problem? How can we screen for this before we get to human trials?
Yeah.
Before there's a human toxicity event?
Well, the focus now is shifting heavily towards advanced in vitro testing, using sophisticated humanized liver models.
Humanized liver models, what does that mean?
These can range from things like engineered microsomes, which are basically purified human liver enzymes like P450s, stabilized in a test tube environment,
Okay.
to much more complex in vitro systems, things called organ on a chip, or liver spheroids.
Organ on a chip.
Yeah, these are like miniature functional cell cultures. They grow human liver cells in a way that accurately mimics the 3D structure and, critically, the metabolic function of the human liver with surprisingly high fidelity.
So instead of relying solely on, say, animal models, which we know often have vastly different P450 systems compared to humans,
Which is a huge problem, yeah.
researchers can essentially feed a new drug candidate into this synthetic, human-like liver environment in the lab.
Exactly. By using these kinds of high-throughput models early in development, researchers can screen compounds. They can see if the human P450 system is likely to produce high concentrations of toxic metabolites from that drug candidate.
They can measure the rates, phase I versus phase 2.
Precisely. Measure the rate of phase I conversion versus phase 2 neutralization for that specific compound. They can start to identify the structural features of a molecule that make it a potential P450 liability. This ability to predict potential P450 accidents early. That's the key. It helps minimize human exposure down the line and hopefully streamline the development of safer drugs.
It's essentially using synthetic biology and predictive modeling to try and preempt a massive clinical failure later on, saving potentially billions of dollars, but more importantly, protecting countless patients from potential harm.
That's the goal.
That transition from just identifying the cause of injury after the fact to actively predicting and preventing it, that really feels like the frontier of modern toxicology.
It absolutely is. The ability to identify early on that, hey, this drug candidate is a substrate for the specific, highly variable P450 enzyme, and that enzyme produces these highly reactive lipid peroxidizing metabolites. That knowledge shifts toxicology from being primarily a reactive field to a much more proactive one. #TAC5, synthesis, and final thought.
Okay, we've covered a tremendous amount of ground today. Really gone from the general concept down to the molecular specifics. If you're listening and trying to synthesize all this, maybe pull out the main points. And the key is that powerful split, the two distinct paths of chemical attack.
That really is the essential takeaway. Just remember those two distinct paths and roughly how they work. Path 1, direct chemical binding.
The immediate assault.
Right. It's fast, it's often localized. The injury is caused by the parent chemical itself. Think of the membrane damage from mercury or the instant mitochondrial poisoning by cyanide. And those injuries often hit the entry and excretion organs first.
Got it. And path 2.
Path 2 is the metabolic conversion. It's generally slower, usually mediated by that P450 system, predominantly in the liver. And here, the injury comes from the body's own essential attempt to eliminate the chemical. The actual weapon isn't the original chemical, it's the hyperreactive free radical or the toxic metabolite that gets created.
Like we saw with CCl4 triggering lipid peroxidation, or high-dose acetaminophen overwhelming that glutathione shield because too much NAPQI was made.
Exactly. So the severity of toxic injury, therefore, it doesn't hinge only on how inherently poisonous the chemical is. It depends just as much on the body's own machinery, our genetically variable detoxification system sometimes acting like a double-edged sword.
Which raises, I think, an important, maybe slightly unsettling, overarching question for you, the listener, to consider as you move forward with this knowledge. As our modern industrial world introduces literally thousands of new, complex synthetic chemicals every single year into our environment, our food, our products, what's the compounding effect? What's the effect of this ever-increasing chemical load on the inherent capacity and the day-to-day function of our P450 systems?
Yeah, that's a critical question.
Are we perhaps chronically taxing the very metabolic pathways that were designed to protect us? And in doing so, are we inadvertently lowering the effective threshold for toxicity, making us more susceptible overall to future toxic events, whether from everyday chemicals or even from necessary medications?
That constant interplay between our level of exposure and our individual detoxification capacity.
Definitely something to mull over until our next deep dive.
Yeah.